Have give us 3 to 5 minutes to make sure everything is. Yeah, thank you for the coordinate. Came through. So maybe my slip, and then I'm gonna just. Okay, so good morning, everyone. Welcome to this breakout session about open science and open for business. So my name is Ted Fawn. I'm the scientific director of Neuro, and it's my pleasure to chair this session. So we have about 45 minutes. Even though we're running late, we're gonna go full for those 45 minutes. And I'm going to start by maybe just showing, yeah, that's one, that's it. And, yeah, so I think this is really important, as was mentioned earlier today during Guy's presentation. We're doing open science because it's a good thing, but I think we're doing open science mostly because we think it'll make things go faster to find new treatments. And this is sort of a chart of how drugs are developed and how industry and academia work together. And as you can see, as at early stages where costs are relatively lower, risk is high. And as drugs move forward through the pipeline, costs go up. And conventionally, the thought is that risk goes down. Is this really the case? As far as neurodegenerative diseases, sadly, we've not done well. And I just wanted, maybe you can click the next. Yeah, so I think this is a lot of the area where we work at here in an academic institution like the Neuro. We do target discovery and we try to find mechanisms of disease. But we can't bring drugs to patients without the help of industry and companies who move things forward. So we're embarking on an open science experiment here at the Neuro, mostly, I think, centered around this part. But what we're going to hear today, I think, is that maybe there's even ways to push the boundaries of open science further along this pipeline, and maybe just the next. So just to show you, and the reason, again, getting back to what we said earlier, is the incredibly high failure rate. As you know, for Parkinson's, Alzheimer's, ALS, there are currently 0 drugs that are disease modifying. And there's an incredibly high failure rate that goes up over 99% as drugs move forward in this pipeline. So we are very happy today to have some distinguished speakers for this panel to give us some really, I think, cutting edge and innovative insights at how open science can happen. We're going to start with a video. And this is from a colleague, very, very close and I think staunch supporter of the Neuro and of open science, Kumar Saikatendu, who is, let me just see if I can read this. He's an associate director in global.
My name is Kumar, and I work at Policy.
Maybe you could just.
We live here on my own, and
yeah. So Kumar is associate director in global research externalization at Takeda. Based in San Diego, he joined Takeda in 2008, and he currently serves as Takeda's teams to leverage external innovations around the world here to benefit patients. And he's trained as a structural biologist, PhD from Bangalore, India, and postdoc at the University of Texas Southwestern in Dallas, and postdoc at the Scripps as well. He's really passionate about innovation in genomics, digital health, personalized medicine, and he couldn't be here today, but he is also very passionate about sending us a brief message.
Or not official Takeda policy. We live in exciting times, right? Times that hold tremendous promise for the future of new medicine discovery. If you look at immuno-oncology, cell therapy, regenerative medicine, RNAi, these were esoteric concepts as recent as 5 years ago. Today, not only are they mainstream, but they have proven very effective as curative therapies. A unifying theme in all of these transformative concepts is the MO of data sharing across academia, hospitals, pharma, government regulatory affairs, all of them working together to develop these new paradigms. Open science, in many ways, exemplifies this, where the main goal, the main emphasis is on getting the science done right and actually make a new meaningful medicines. I think global scientists, on a more serious note, share a collective shame that diseases like Alzheimer's and Parkinson's and others, that despite being around for 100+ years, not only can we not treat them, we actually don't even understand these diseases, what causes them, what's the underlying mechanisms, and so on. So I think open science today is a movement that's largely driven by necessity more than anything else, but also this humble realization that we have to try something out of the box in our quest for discovery of new medicines. I'm an eternal optimist in the collective intellect of the millennial generation and wish them well as they share more and collaborate more and come up with the next cure. So good luck.
Great. So I think that's a fitting kickoff of this breakout session, and I just want to emphasize that large multinational pharma like Takeda have, despite the idea when we started this that industry will run away from anything if they can't patent and protect their investment with IP. They've invested quite heavily in our drug discovery platform that's being led by Tom Durkin sitting in the back there. So actually, are we starting with Diane? Maybe I think it would be better to start with Diane if you don't mind. Do you have? Could we put? Yeah, I put it on the thumb drive as well. We switched it. So Diane is an accomplished scientist and leader. She's been involved in financing of innovative biomedical research for over 20 years. Since 2012, she is the president and CEO of CQDM, the Centre Québécois du Médicament. And she has worked in the inception of CQDM when she joined in 2008 as VP Research and Business Development. So she holds a PhD in microbiology and immunology from the University of Montreal, as well as an executive MBA from UQAM and from Université Paris-Dauphine. She's worked at, before joining CQDM, at the Fonds de Solidarité, FTQ, where she managed an investment portfolio of $40 million and helped several companies implement their drug development strategy. She also sits on a number of boards, including Montreal in Vivo, CIHR, Ontario Genomics, Catalyst Québec, CQDM, and Institut National de Recherche Scientifique. How's that going?
You said it's on the thumb drive?
Scientifique. I mean, that's what you sent me. I don't know if you have. I don't know if you sent me something since then.
Oh no, it was just one slide. I sent the whole presentation to
Helena.
Yeah.
So.
But you have any? Yeah. I can put this on mute.
I'm sorry. Yeah.
You want to start with the others?
Okay, let's start with So after that long introduction. So Owen Roberts is the Chief Executive Officer of both Nobelex, Biotech, and M4K Pharma. M4K Pharma is a company dedicated to discovering, developing, and commercializing affordable new medicines for children's orphan diseases through open science. Quite innovative. He's partnering with academics, foundations, and industry groups to fund its drug discovery and drug development programs. M4K uses the open science principles of data sharing to encourage research collaboration and to reduce the cost of its programs. Prior to founding M4K in 2017, Owen was co-founder of Affinium Pharmaceuticals, a privately held clinical stage antibiotic company. At Affinium, Owen was CFO and led the company's business development until the successful sale of Affinium to Debio Farm International in 2014. So relevant to the Neuro, M4K has also been a model which is under the umbrella of the Agora Charitable Trust that has inspired the creation of the M4ND, which is dedicated to neurological diseases. So we're quite excited to have Owen. Thank you for coming.
Thanks. I'll try and keep my presentation shorter than the bio. So I'm going to do this in about 10 minutes. There's a lot of stuff I'm going to skip over, but I'm really looking forward to the questions. But really, at the beginning and end of the day, M4K Pharma, is the personification of an experiment in open science as a commercial company, and to actually take open science and move it down the pipe into commercialization. So starting from the very beginning, science has been outpacing the historical business models that we've been used to. Most of the people here in this room, this is your fault. We've taken very large diseases and you have subdivided them into smaller and smaller diseases. Something that used to be generically just called a brain tumor is now 100 different diseases, all these things. And so now we, instead of focusing on a neurological disease or a tumor or anything else, which was carpet bombed with one set of therapies, is now subdivided into smaller and smaller and smaller diseases. The problem that becomes here is what was once a large market, or considered a large market, maybe a very crappy therapy, but what was once a large market, it's now actually a very small market, because now you're targeting very small groups. And now what we've done is we've created a whole series of orphan diseases out of once was considered a very large disease. So the problem becomes, does our business model actually work for this? Because as the size of the market has gone down, the costs have not gone down. In fact, you can make the case that the costs have gone up because there's more work being done. It may be harder to recruit the patient population. Clinical trials may be going longer. There are a lot of many other things that are going there. But hopefully, at the end of the day, the work that you've done by narrowing down the diseases means that at the end of the day, a better therapy will be applied against the right disease. So what we're having here is the cost of patients is increasing. And what does the current business model do? Excuse me. You have a smaller market, you charge more. This is all across the press these days. You see it all the time. $300,000 a year per annum for a therapy treatment. This is stressful to the patient population, the healthcare system, and ultimately governments, who at the end of the day do bear the brunt because they are, at the end of the day, us. So can you develop effective therapies into smaller markets? So this has become the question. We've had this whole one business model for the longest time. And we've had the square peg in the round hole. Or conversely, we've been trying to put a round peg into a square hole. I like that one better. That's generally what happens. So what we've done here is, can you use open science to develop and commercialize drugs? Not just the early stage academic work, but moving it down the chart that was put forward earlier at the very beginning, which showed all the work going further down. Can you do it to make small market drugs more markedly available? And a lot of the work that you're doing by subdividing the diseases, you are creating more of these submarkets. This problem is not going to get better, it's going to get more problematic. So, first off, everyone talks about open science. I think different peoples and different groups have different interests or different definitions. I have a bit of a commercial view of it. It's a commitment from the company to rapid multilateral sharing of knowledge, results, data, everything without any patent restrictions. And in our world, that patent restrictions is a very different concept. It may not be the important part in your world, where it might be papers or the institutions, rules, etc. But a large part of it in our world, in the commercial world, is we have lived off patents. Patents have been the driving. The reason for patents is, if I'm going to raise money or do something, I have to own something. That's the capitalism. I have to own something so that I can then sell it, use it to make more money. The way of owning something intellectually is to create a patent. So fundamentally, that has been the tool for the historical model, to create a patent, to have that knowledge base and go from there. That has been the historical way to do it. With open science, as you can see, this doesn't really work initially well with VCs or other usual ownership stakes. We've been using funds from foundations, granting agencies, corporate endowments, academic institutions, and that's endowments, but I actually meant corporate donations. And this latter one has actually been a big plus. I'll get into that in a little bit. Commercial opportunities do remain. If discovery developments are reduced, sustainable pricing can be achieved lower. And I'd like to also put out something that people don't realize. I'm going to spend a few minutes on this. There is two worlds out there. There's the patent world, which is 20, 22 years of I own this. But then there's a regulatory asset. The regulatory is what the government and jurisdiction, the FDA, Health Canada, EMA in Europe, allows you to sell this drug. So there's three things that I always say. Well, a lawyer told me this. So 3 things you can guarantee you're going to get in life. You're going to get a Christmas card at Christmas. You're going to get a birthday card at your birthday. And when your patent expires, you're going to get a letter from Teva saying that we think your patent is useless. Well, actually, when your regulatory approval expires, Teva's going to come after your patent. Regardless of if your patent you think is valid, you may have 6 years left in your patent, the generics are going to come after you after your regulatory approval expires, because they can't do anything about your regulatory approval. So, what did we do in our experiment? Well, as I said, this is an experiment, and this is the first one we've done. So we picked a disease that represents a very urgent medical need. It has a small, limited market. There's not much competition out there. There's no one pressuring us from it. We picked one with a known genetic target. Why? Because our company and our group is full of people who understood genetics. So we started with the science that we knew. And as I said, they had insight into the target in this case. And it also has a favorable clinical path. What I mean from that in this case is because the disease is small, the regulatory bodies will allow you to get registered without going to phase 3 because it's just too small. They may allow you to get registered with maybe 20 to 30 patients. And so therefore the cost is low. The patient population is well organized, so recruiting is not a problem. Shouldn't say not a problem, but recruiting is easier. So these things are. So our objective to access grants, foundations, and corporate partners, and to meaningful and aggressively share and receive scientific insights. We demand in all our MTAs that everyone shares. It's not good enough, and we've gone through this, to say, oh, we're not going to put a restriction on you. Well, then you send it off to a university, and they have their restrictions. We make sure that any partner we work with, their institution also removes those restrictions. Because otherwise, all you've done is move the roadblock down the road a little bit. So it's very important when you do this to make sure that you actually do have an MTA. But the MTA is an unusual one, which says there are no restrictions on it, and you will not put a restriction on it. That's really important, because their default is that they're going to put a restriction on it. We aggressively promote open science. Sometimes I attend symposiums and give talks. But we also actually put everything out on our webpage, on our blog. Every month we have our standard project meeting. We go through the cascade of compounds we've developed. We show the structure. We say which compounds worked in the initial assays, which ones got kicked out, which ones went to the secondary assay, which ones got kicked out, which ones went into animal for PK. Blah, blah. It's all every month, and they're posted on our blog. You can go to our blog, and you can hear my whiny voice go through every time and ask stupid questions, because I'm not the scientist. But we go through that, and we find motivated partners. Again, the benefit of the disease we chose was you do have a motivated population, and I'll get into that later. It's unfortunate, but with the motivated populations, people also approach you, and also other corporations will approach you. And in fact, people who've looked at it and failed will approach you because they want this to succeed. Key is we retain IND data, investigational new drug data. This is the file that you send to the FDA or Health Canada. And this data is something that we can retain. We will show all the information out there. But at the end of the day, it's still ours. We are the sponsor of the clinical trials. This is our data. You can use it. You can do your own research on it. We'll send it out to you. You can look at all the information. But that data is ours. And when we put an application for a new drug application, the sponsor is M4K Pharma, and the regulator will only accept that data from us. They will not accept. If you want to use the data, you have to repeat it and do it yourself. So you go all the way back to the start and do all the IND enabling studies. So the disease we picked was diffuse intrinsic pontine glioma, DIPG. It is a terrible disease. It's a pediatric brain tumor. There is no treatment for it. There's about 100 to 200 patients in North America a year who get this. Basically, you have a very healthy child, and then all of a sudden, somewhere around the ages of 8 to 14, the child trips, they fall, their coordination goes, their eyes become cross-eyed. You take them to the doctor, and the doctor tells you your child's going to die in 12 months, and there's no cure. And it's crushing, and the families, we get about one email a month from some families saying, I hear you're working on this, can we try your drug? They're desperate for something. They're reaching out, they have, and it's very sad. I'm not going to get into much more, but as you can see, it is a very, it's a failed disease. The survival rate is non-existent, really. So, we created a company. Why use a company? Well, because we can be a unit to get grants, sign contracts, do the work, outsource with collaborators, move things together. But at the end of the day, what we're doing is we are a virtual biotech company, and we're taking all this data. I've done drug development before, so have my other colleagues, and we're taking this and we're marching it down the usual path of getting a regulatory approval. We're going through all those processes. We're not trying to do this as, an academic thing where, this is interesting, I'm going to hop over here. No. If this isn't, it's interesting, someone else do this. Give me another drug, and let's put it into this molecule. Give me another drug, okay? I don't necessarily care why the mouse died. The mouse died. Give me another compound. Find one until the mouse works. Mm-hmm. Why the mouse died, I'll leave that to someone else. I'll put it on the paper. Someone else can follow that up and find it. I'm marching towards an IND filing. And it creates a vehicle where I can enter agreements and do other things. So what does the corporation give me? No, I have a ton of flexibility. My parent organization, in this case, is in fact a Canadian charity. So if someone is a charity and can only donate to another charity, they give the money to our parent and they direct it down to us. If someone else wants to do a contract with us directly to make a relationship, a business relationship, they can give it to us. So we have flexibility. That's why we picked the corporation. That's the main reason. So differences between a normal biotech and M4K Pharma. You have public or private shareholders. We have a charity. What's your asset? In a normal biotech, it's patents, licenses, etc., all those kind of things. Ours is the patents. We do have licenses and contracts. We have entered as a corporation into those various agreements. But the biggest thing that we have is the regulatory exclusivity period. And so what I'm going to show you very quickly, and this is the key. I'm sorry if I'm doing more than 10 minutes. I'm going to try to. So, year 0 out to God knows when. You do a little science, you file an application, 1 year later, the patent application, so you do it, you put the provisional patent application is filed, clock starts ticking now. So you got 20 years plus some extensions. Issued, this is when your patent is issued. Meanwhile, you're doing all this work, you finally get to, let's say, year 10, as from your chart, your IND gets filed, you do your NDA, you've done all your studies, your clinical trials, etc., and then a year later, NDA is approved. So you're about year 16. Okay, so from there on, you have about 4 or 6 years to go. In the U.S., if you have a new clinical study for a new clinical entity, you get an exclusivity after the NDA is approved of 3 years. Plus, if it's an extension for pediatric studies, you get another 3.5 years. Okay, if it's for just for the clinical studies. If it's a new clinical entity, i.e. a new compound, you're now looking at 5.5 years. And by the way, no one can attack me during this period. No one can come to me and say, you know what, someone presented this in Timbuktu in 2001, your patents expired. We discovered this. oh, that's not really novel. Oh, it was done somewhere else. No, I have exclusivity. The government has told me I'm the only person allowed to sell this drug. So I have a real asset given to me by the government. And again, if it's an orphan drug, I know it's 7.5. Look, I'm past my basic period of a patent. This is more valuable than a patent. EU orphan drugs, you're out there. EU new chemical entities, etc., all the way down. But the point is, as you can see, this is something that's very valuable. So I don't need the patent. I can share the initial information. I can get people to work with me. I can lower my costs. I have a whole slide on finance, but I decided this crowd might not be the finance crowd. Save it for later. So what are we asking now? So from the funding sources for normal biotech, you have VCs, equity, etc., people who want to get some of the cash flow out of the company. Here we're looking at grants, foundations, and philanthropy. There's another key. I'm sorry, I'm going to really run through this fast. Yeah, but there's a key. Jane, in the earlier presentation this morning, made the comment, no, these people want a drug. If we get a drug on the market, they're going to be happy with us. That's what they want. They don't really care. So there's a key. There is a risk. In a small market like DIPG, there's a very small risk that someone's going to come along after us. Why would they? There's no incentive. There's no commercial incentive. If we were going for a large neurodegenerative disease, where there is a big one, we may run the risk of what I call the Tour de France syndrome. We're the breakout pack at the very beginning, and the peloton just sits behind you, sucking in your air. And then all of a sudden, about 500 meters from the finish line, the peloton swoops by you. The peloton could be the large Pfizers, etc. If I was working on neurodegenerative disease, I'd be very disappointed. But my funders, they just want the cure. They don't care. So the fact that Pfizer or another pharma comes on behind and actually gets the drug in the market, they won't mind so much. And then we want to be uncomfortably open. I know I'm running out of time. These are the sources of funding. I just want to point out one thing. In phase 2 and registration, there's a lot of money coming from advocacy groups, etc. But there's a potential for investors as well. There might be a potential for investors at that time because you are close enough. You have de-risked the situation. You have raised most of the funding, and you have dropped the costs. So how's our experiment going? All these parties have joined us. We have granting agencies. We have companies that have joined us. We have other institutions, universities who have joined us. And universities are really important for this because we are dealing with a drug which not too many patients have. So guess what? Love Charles River. They're being very helpful for us. They don't run a model for this. Why would they run a model when there's no one to serve? The academics run models because they're interested in it. And so we go from there. How's it going? Well, we've actually synthesized over 300 compounds a day. We have 5 that have reached our original target product profile, and they're moving on to candidate selection. Our monthly projects are going up forward. We've had wonderful support. We've been contacted by people on the phone at these meetings who said, by the way, we ran a program, it ended, here's information that we've learned from it. And that came from 2 corporations that were on the phone and did that. So we have to repeat the experiments. As I said, you have to be able to do it. So many other opportunities which meet the DIPG profile, dire unmet need. Most neurological diseases are dire and unmet. The whole issue around Alzheimer's is terrible. The disease doesn't fit the historical model. As being said, Parkinson's we kind of know for 100 years, still haven't figured it out. The initial technology is derived from experts using one of the parts. There's a lot of base technology around neurological diseases. So I think there is an opportunity for M4ND to work on this and become part of it. And there are partners that we can work from that. So anyways, I'm going to look forward to hearing your questions, etc. I'm sorry it took more than over 10 minutes.
I think you were fine. I think we're just running in general late, so we're going to try to keep things, and maybe you can save the questions and discussion for the end. So, Diane, I will pass it over to you.
Thank you. Thank you. So, thank you for the invitation. So, I'm from CQDM, as you know, CQDM's mission is to accelerate the drug discovery process. So we want to support tools, platforms, or any kind of research tools that can really accelerate the discovery and the development of new drugs. So what we want to do, if we want to reach this, we are absolutely convinced that we need to break the silos in research to speed up the process. So open science is one way to do that for sure, and I'm going to provide some examples of this. I'd like also to discuss a bit of open innovation and stretch it to, in fact, as many models of collaboration that we can imagine, because this is what we have done at CQDM, especially with the pharma industry. I think that we need to find new models and new ways to collaborate. And as long as you speed up the process, as long as you find ways to generate some mutual benefits for everyone, I think that you can do a lot in research, and hopefully I'll provide you with the right examples for this. So just to start with, to give you a little bit of information about CQDM. So CQDM is a non-for-profit organization led by the pharma industry. So we have 13 big pharma companies who have joined CQDM, put money into CQDM together with the Canadian and the provincial government. So I mentioned about our mission. Another piece of our mission is to make sure that we are going to create a collaborative ground so that we are efficient in accelerating the drug discovery process. Our collaborative model is really based on several different aspects of collaboration. So, number one, multidisciplinary, for sure. That makes sense. Multi-institutional research, networks of different institutions working across Canada together. Public-private partnerships, so the pharma industry, the biotech sector working together with the academic institution. But probably more importantly, for me at least, is to see big pharma companies working together in collaboration. They are not supposed to work together, they are supposed to compete. But what we have done in CQDM is provide incentives so that they can start working together as well. So the model is creating, this collaborative model is creating a financial leverage that is very, very important if you want to de-risk early stage research. So in a nutshell, the way it works is that the pharma companies are putting money into a project. Sometimes you have, in most cases, you have several pharma companies sharing the cost of the research together with the two levels of government and some co-funding partners. So you mentioned a lot about foundations. We do have a lot of foundations as partners who are helping us to support the research. So it's a pool of money that we use to support a given project. And because we have that pool of money, we create leverage that is very important, at least tenfold, so that we can afford to de-risk the research, to work early stage, to do some pre-competitive research, and to afford to do to support the kind of research that you were talking about. Just one word, CQDM is pan-Canadian. It's a pan-Canadian consortium, so we are, of course, very active in Quebec, but also outside Quebec. So far, we have supported mostly 80-ish outstanding projects for a total of 100 million. And this is creating a lot of platforms, as you can imagine, research tools, but also over 150 therapeutics that are currently being developed. So we're very proud to see that this is leading to drugs, and this is, what all this is about. Now, a few examples of collaborative models. I'm going to start with someone that you know very well, Diane, who, we have supported this project, I think, a few years ago. This is a collaboration between CQDM, Brain Canada, and two big pharma companies. So in this project, I should say initial project, because you had several other projects that were derived from this. what you Vlad wanted to do is to create some iPS cells, so generate iPS cells from PD and ALS patients, so that you can generate a platform for drug discovery. So all the results, the cell line in the assays, are put in the public domain. But still, this is of interest for two big pharma companies here, because what mainly the incentives here is that they want to work with KOLs. They want really to work the best investigators in that field, interact closely with them, and they see this as a real benefit for their own organization. Number one. Number two, well, even if everything is going to be put in the public domain, they are going to have an early access to the results because they are interacting with the investigator, and even that is still for them a great benefit. Now, after this first project, we have seen a lot of different initiatives being generated that are also of interest for pharma. So the creation of this drug screening platform with Neuro initiative, with the additional funding from Merck, from Takeda, who also use this platform to screen their own library of compounds and create some drug candidates. So as you can see, there's a lot of benefits that you can generate for the group of researchers, but also for pharma companies. And this is what I want to illustrate here. Now, another project that we have supported at CQDM with the same principle. So it's a project led by Cheryl Smith from University of Toronto with SGC. Here we have two big pharma companies, Takeda and Pfizer, who are funding this project. And again, the same principle. All the results are put in the public domain. And the reason why big pharma companies are joining this project is really because they want to be close to the best investigator in the world in that field. And they want to interact with them. And if possible, maybe influence some of the assay development so that it's going to be aligned with their needs. But again, everything is put in the public domain. Everybody can benefit this. Now I want to switch a little bit to a different type of collaborative model. Something that I would call maybe more open innovation model. So in this case, we have supported the development of a library of microcycles. So it's really a library of molecules. So pretty difficult to share with the pharma industry. So in this specific case, we have 4 pharma companies. I have one missing here, I'm sorry. So AstraZeneca, Pfizer, Merck, and GSK, who funded this project. And what do they get in return? Well, they get, first of all, I must say the IP is owned by Encycle in this case, which is a biotech company based in Toronto. So in return, the pharma companies would get a right to screen this library with a few targets of their choice, and it's non-exclusive, so other pharma could come here and screen this same library, but they get to do that a little bit faster than the others. They also get, again, the opportunity to interact with a key opinion leader in that field, and they feel that this is a right incentive for them. In this case, all of the 4 pharma companies have exercised their right. They have used the library, they have screened it and found hits. And then if you find a hit, then you need to sign or discuss an agreement with the IP owner. So in other words, you're pushing the tough discussions for later in the process once you know that there's a value for this given company. So it's another way of looking at this to speed up the research to a point where you are able to say, well, this is of interest, and then we need to sit down and look for some ways to share the IP in this case. My last example is an example from Quebec. So it's a collaboration between Frédéric Lalonde at CHUT Québec and David Gay at Feldan Therapeutics, a small biotech company based in Quebec. So in this case, we have 5 big pharma companies who are sharing the cost of this research. So this is about developing a delivery system for intracellular delivery system for peptides and antibodies. So again, in this case, the pharma will get a non-exclusive right to test, to use this platform and to use for research. So they are going to use the platform with their own molecule to up to a certain point. So in this case, it's the preclinical proof of concept. So if you reach that point, then you need to find a way to agree with the IP owner, in this case Feldan, to future development of this technology. So it's something that can generate benefits for Feldan, as you can imagine, because you not only validating the technology, but you're showcasing your technology to 5 big pharma companies with the potential of having partnership with big pharma companies while you're developing your own technology. So I'm just trying to bring you a few examples of how we can work with pharma and generate win-win situation for everyone. We have at CQDM a lot of these examples. So because we had so many projects and so many collaborative models, we could generate a network of people who are willing to work together. And I think that's also another way to, it's about open science, but it's also about sharing the results in many different ways. So it's possible to work with pharma, and what I like to see is them working together as well, sharing the cost of a research and being there early in the beginning of the research so that we can speed up the process. Thank you.
I think that you had two very sort of different examples of how open can work with industry and with business. Something I think, Diane, I remember at the beginning, it was very. I think that CQDM has moved a lot in the open compared to maybe even 4 or 5 years ago, and I think your examples are great. And then there's something completely. Sort of off the charts here. That's a real experiment. That's in a real life experiment. We have very little time. I apologize. I think that we're trying to stay on time. But I think if there's like a few questions, it would be a good time to.
Owen. Yeah, my name is Mark. So your experiment is really interesting. But neurological disease. So let's talk about M4 D.
Yeah.
I'm not sure, and I don't know if Ted would agree with me, that small molecules are really the way to go. So we're more talking about cell therapy, gene therapy, viral vectors, modification of cells, and there's a lot of patents around all that space. And you want to use a type of viral vector, you need a license and all this. Have you thought about this at all? What are your Maybe not. Maybe the experiment is easier done with the small molecules, but where would we be going with this M4 and D in a broader space in small molecules?
Yeah, so first off, your point about the small molecule is correct. I mean, if you take a look at what we did with DIPG and that disease, we selected it with the greatest chance and the fewest impediments. Sure. So that's that on the table. Yeah, that's going to be a problem. There are places where there is a large space of IP owned by other parties can be problematic. There is still the space that what are the holes inside that space? I think my understanding of some of that IP, not all of it, but is that there are a lot of people kind of saying, well, that might be hard to actually enforce some of the IP that's around this because it's more of a scientific know-how rather than an actual intervention or something like that. So without getting into it, that will be a problem. But at the same time, as you take that basic science and move it forward, you can be creating new know-how and aha moment or a type of invention. And if you put that on the public domain, you can say, this is based upon this invention, and therefore it is either an advancement or it's different from your invention. Not saying it's not a problem, but that is.
I would like to add that
Yes,
I think the jury's still out on that.
Okay.
I think that for sure that biologics in general are very promising, but I'm not sure. As I said at the beginning, we have 0 treatments. still don't understand very basic, mechanisms in these diseases. So I don't think I would put all my eggs in one basket for sure.
That's what I was saying, but.
One of the comments I have regarding, you mentioned about